is Paula Reyes Herrera, and I'm going to present you the work First Genome Assembly of Cultivated Sweet Granadilla, Passiflora ligularis. I'm a researcher at Agrosavia, la Corporación Colombiana de Investigación Agropecuaria. Well, Colombia is the leading producer and consumer of sweet granadilla, Passiflora ligularis, and it is the second most economically important species in the Passiflora genus for Colombia, and Colombia has an important diversity in this genus. It lacks a genomic reference, and this causes a limited understanding of its genetic variability and hinders its potential inbreeding efforts. And this study presents the first genome assembly for this fruit, aiming to facilitate the genetic research of this species. Here you can see in the image, you can see the fruit and the flower of Passiflora ligularis. So for the methodology, we had an accession from the Colombian Gene Bank. We extracted high molecular weight DNA and we sequenced 3 high-fired cells. We estimated genome size, heterozygosity, and ploidy based on reads, but we also used these reads with 3 genome assemblers. And in parallel, we search for genomic resources for the, available in public databases for the Passiflora genus. We found 3. We compared this genomic reference based on quality and proximity to Passiflora ligularis, and we selected a reference genome. From these 3, it was one of Passiflora dulis. We used this genome assembly and the assemblies that resulted from these 3 algorithms. We use this data and use the software RagTag to do the scaffolding. And then we compared the contic level assembly and the scaffold level assembly by using several metrics. And we selected the Passiflora ligularis genome assembly. Distribution for these reads. From this estimation, we found that 20x coverage, a genome size of 1.28 gigabases, and a diploid genome was confirmed with evidence of genomic duplication events. Moreover, here in the table, you can see the comparison of several assemblies from the 3 algorithms with at the contic level and scaffold level. The one highlighted in gray was the one that was selected. It was selected because the total length was the closest to the estimated length. So the final scaffold assembly yielded an M50 value of 165 megabases, comprising 9 scaffolds and a total length of 1.45 gigabases. And here in the figure, we did a comparison between the selected assembly for Passiflora ligularis and the reference genome of Passiflora dulis. And we found similarity between both assemblies. Moreover, as an interesting result, we found that the proportion of repetitive elements is high, is between 86 and 87 for Passiflora ligularis, but also for Passiflora dulis. The Lie score, that is the LTR assembly index, was 14.22, which reflects a reference quality assembly for the Passiflora ligularis assembly. This study presents the first genomic reference for Passiflora ligularis, and well, we found that the repetitive content is high. However, it aligns with that of Passiflora dulis. As future directions, efforts are underway to generate RNA-seq data to annotate this genome assembly and to identify genes that are unique to the species. Thank you very much for your attention.